Hello, my name is Michelle Miller, and I am a scientific officer at Bioanalysis. Thank you for joining today, where we'll be talking about the strategies for long-term success, specifically streamlining the preclinical to clinical assay transition. To begin, I'm going to give a little bit about Bioanalysis as a company. So we are a CRO that specializes in bioanalytical assays, specifically the assays that you see shown here. There's a broad array of buckets of assay services that we offer. The presentation today is really going to focus on what you see in the very first column here, which are immunogenicity assays and PK assays. But one thing I do want to point out on this slide is that when we engage with our clients, these assays are offered across the full spectrum of the drug development process. So sometimes we're engaging very early at the discovery phase. Other times we're engaging with our clients when they get closer into the IND enabling preclinical and the clinical phases. And we even have clients that stay with us all the way through post-marketing manufacturing support. So the full spectrum of drug development. So it's really what's going to drive the discussion today is our experience working with these assays in that preclinical to clinical sweet spot that you'll see right there in the middle of that spectrum. So I'll dive right into the content. What I'm going to give you first is an overview of the preclinical and clinical assays and what assays transition between those. I'll give you some examples. And then we're going to go into more specifics, and I'll break that out by assay type. So I'll have a section that focuses on PK assays, and then another section where I'll focus on the immunogenicity assay strategies. As a backdrop to this discussion, I did want to show a little bit of information around that transition of a drug from preclinical all the way through approval and focus more on the time and the money that's invested in that. So this slide shows some data from a recent publication that went back and looked at drugs that were brought to the market in the last few years, and they showed that in 2022, the average cost for bringing a drug to market was $2 billion across all these phases. And you can see that broken out on the bottom here in blue. And that this whole process took about 14 years. So obviously a lot of time, a lot of money going into bringing these types of medications to the market. But then add on to that about 90% of drugs that enter this process fail once they reach the clinical trial phase. And this can be for a variety of reasons. It could be for issues with safety, issues with efficacy, but whatever it may be, only about 10% of drugs are successful despite the amount of time and money put into them. And so what's really critical is being able to identify areas where you can save time and money on some of these drugs that are not moving forward, and then use that additional time and money and roll that into the ones that are being successful in this process. And so that's one of the things that we really want to hit upon today with these bioanalytical strategies is that testing is a very small part of the overall budget when it comes to preclinical and clinical phases, but it is a very important part, and there are key ways that you can leverage that transition or streamline that transition from preclinical to clinical in order to help save the time and money. So as I mentioned, I'm going to show you a very broad perspective of the assays that are used. This is an example of an antibody drug conjugate and the overall assay strategy at each stage. So you'll see starting on the left, these are the non-clinical or early preclinical tests that are done. And then as you move towards the right into the IND enabling non-clinical and then into the different phases of clinical trial testing, some of those assays that are listed below those stages are going to shift a little bit. But what you can focus on are the similarities, and that's where I'm going to break out the next two sections of this discussion. The first is the pharmacokinetics. So PK assays are a critical component for any drug program. And in this instance, you can see the exact same types of assay strategies that are used for the PK measurements are used starting at that IND enabling preclinical and continue all the way through clinical. And then the same thing for the immunogenicity assays. So now we're talking about anti-drug antibodies or neutralizing antibodies, ADA or NAB assays. And these start to be implemented in that preclinical space, but will also be carried through into clinical. So two key areas where you can see similar assay strategies and you can focus in on making those transitions from the preclinical to clinical phase smooth. So starting with the PK assays. I'm really going to focus again on that IND enabling stage. So this is when you're getting closer and closer to being able to move into your first in-human studies. And we're going to look at PK assays here and then compare them to what you would see at a clinical stage for the same therapeutic. There are a couple of high-level bullets here, and then I'll go through each of those in detail on the following sections or the following slides. These are key areas that you can focus on when you're comparing the preclinical and clinical stage. So the first is the assay format. So I'll talk a bit about what is used at the preclinical, what's used at the clinical, if they're the same, if they're different. Then we'll get into dosing concentrations, so the difference between what you're doing at the preclinical stage and then what you're dosing when you get into your human studies can be vastly different, and that can impact your PK assay. The other is matrix effects. Obviously, moving from animal models into humans, you're using a completely different matrix, and there are a number of components that can impact your PK assay there. And then the final thing that I want to talk about in more detail will be the drug lots. So the actual manufactured lot of your drug material that's being used and being measured in that PK. So starting with assay format. There are strategies around preclinical PK assays that can produce a generic method. So these are PK assays that are built to detect something like a human IgG antibody inside an animal model. So as you can imagine, if you're putting a human or humanized antibody in an animal, there are no other antibodies of that specificity in that animal. They only have their antibodies. And so you can target those unique sequences for the human. So you can have reagents like anti-human IgG-Fc that are used to capture and detect that therapeutic. And because you're targeting that constant region of the IgG molecule that's a human or a humanized version of IgG, you can actually use this across multiple programs as long as each of those programs has an antibody therapeutic. So specifically for monoclonal antibody programs, this is an excellent tool that can be used, but it is only specific for your animal models. So you can't use that once you move into the clinic. So it's worth mentioning there are a number of positive aspects to using these types of approaches. So they're very quick to implement. As I said, you can use them across different programs if you have more than one antibody therapeutic program. It's a very efficient workflow once it's established. But the drawbacks are now you have an assay that can be used at this stage, but it's not going to be appropriate for the clinical stage. It is modality specific, so I am talking about antibody therapeutics versus all the other modalities that exist where you can't take this strategy. And then there are some differences between your matrices still that you have to work out. So maybe a generic assay, you could still use the same format, but sometimes you'll run into issues when you switch from one animal species to another, for instance, where you do have to go back and tweak the assay a little bit. So it's not always completely hands-free once you have it established. So worth mentioning that this exists. It's a very particular application. And in the context of this discussion, if you start with a generic assay, then there isn't really a good way to streamline the transition. You're still going to have to go all the way back to assay development and create a new clinical assay. So then this slide is showing you the various methods that are appropriate for starting in the preclinical stage and moving into the clinical stage. There are a variety of ways that you can build a specific PK assay, like what's shown here. You can use the drug target to capture or to detect, or you can have antibodies specific to your drug that can capture or detect. A number of methods with which to use specific reagents to build the assays, but what I'm going to talk about will just be a general summation of all of these together. So the things that I'll suggest will carry across no matter which format is being used. And the benefit, of course, of building a specific assay very early on is that this is a format that is appropriate at the preclinical stage and the clinical stage. So it can support all phases of your molecule. Once you have it established, there will be some tweaks and things that you need to do to the assay as you move from one study to another, but it is an overall reduction in the assay creation burden. So the amount of time, the amount of money that has to be put into the assay is greatly reduced at each of those stages. And then, as I mentioned, it's appropriate for the clinic, so you can have a quick implementation of your assays when you move into the clinical trials, and you don't have a lot of time eaten up by assay creation at that point too. The drawbacks, of course, are that if you have a specific assay, you need specific reagents. So there is a generation of reagents part of this that requires a monetary commitment, and it's something that needs to be implemented with enough time to have the reagents available. For example, developing an anti-id antibody against your drug compound is going to take months. It's not something that's really quick and easy to procure. But you need several months built in. It requires a certain amount. of cost associated with it to get that generated, and then only once those reagents are available can you build this type of assay. The other consideration when you're looking at your preclinical assays and thinking about what it's going to look like when you move into the clinic are the dosing concentrations. So let's say you start with a specific assay, you're building it in a mouse model, you're going to use it for NHP as well, and then eventually you're going to use that same assay format in humans. If you look across the tox assays listed here with a single dose in mice, a single dose in an NHP species, you could see that the serum concentrations in these cases for the two animal models are actually pretty close. This is from a publication that's available through that link listed at the bottom, but they published each of these sets of data so they could be compared. So they're pretty similar if you're looking at the y-axis. Then when you move over into humans, you can see there are log differences in the amount of the drug that is supposed to be detected by your PK assay, both at the upper and lower limits. So the sensitivity of your assay, even though it may be appropriate when you build it and use it in your animal models, when you move into the clinical samples, that may no longer be appropriate. So there could be some adjustments needed if you don't have an assay that's already built with the appropriate LLOQ at the preclinical stage. And then finally, the other big difference that you can have are the matrix effects. With building a PK assay, there are some very early experiments that are used to set the stage for how your assay is going to perform. And that can have to do with reagent concentrations, the way you're handling your samples, and MRD. One of the things to keep in mind for assay development of a PK is that when you focus on some of those parameters and improving those, it could be at the sacrifice of other key parameters of performance. So in this image, you'll see on the one side, there is the signal and sensitivity of your assay. On the other side, we have selectivity and specificity. If the focus is really driving down the sensitivity, getting a really sensitive assay, things that can be implemented are reducing the MRD, having higher detection reagent concentrations, and those will improve your assay performance when it comes to the sensitivity or the signal window of your assay. But at the same time, if you're taking those tactics to try to drive down those parameters and get those within optimal performance, you may be sacrificing selectivity and specificity. So a more sensitive assay may be more prone to specificity or selectivity issues. And then vice versa. If you're really focused on improving the selectivity and specificity of the assay, some of the strategies there are to modify pre-treatment strategies for your samples, to improve the blocking protocols so you have more stringent blocking, and then to alter the MRD by increasing the dilution of your sample. So all of those things can improve those parameters, while at the same time, it will potentially drive the assay to be less sensitive. So the key here when you are developing your PK assay and thinking about how all of these things work together is to find an appropriate balance. And the strategy that has helped us implement the appropriate methods in order to optimize the assay, not just for preclinical, but also for the future clinical studies, is really to get in there and do this very early on in development at the preclinical stage and include matrices from both your animal species that you've selected and human matrix. So being able to evaluate all of those matrices very early in development will get you an idea of where your assay is performing across each matrix. Now, the final consideration for something that can help or hurt your advancement of an assay from preclinical to clinical are the availability of the drug lots. So production of a drug or the manufacturing of a drug is a dynamic process, and it starts early, but along the way as you're testing your drug, you continue to have improvements in the manufacturing process, scale up that may change the way that your final product looks or performs. You have multiple iterations of that drug being produced and released in these lots, and there can be lots differences. One of the keys to having the availability of hopefully a final lot of drug is to be able to address any of those issues early, looking across multiple lots, looking at any potential changes, and really trying to predict if the assay is going to perform differently. And I have a couple examples here of how that's going to impact your strategy. I'm referencing the ICH M10 document here, where very specifically it outlines the expectation for the lot of drug that you're going to be using in a PK assay. And it is expected that in the PK assay, your standards and QCs are derived from the actual drug that is being used for dosing. So that means if you start with a GLP tox lot of drug when you're building your preclinical assay, then that's what's going to be used in the assay once you're analyzing samples. So you can validate with that lot, and then you can move into sample analysis with it. However, when you move into the clinical side, you're going to need your clinical lot of material being used in the assay. So that does impact your strategy, right? If you have everything available before you ever get to the clinical trial, then you can go ahead and you can validate your clinical assay very early on using that lot, but that almost never happens. Usually you have your GLP tox lot available, you have your full evaluation of that in the PK assay, you do your validation with that in the PK assay, and then you analyze your GLP tox samples. Then at that point, you have to make a decision. If you don't have the clinical assay lot available, then you can go ahead and validate in your human matrix using the GLP tox lot. And then you'll have to go back and bridge in the clinical lot later into your validated assay. The other option is that you can wait until you have that clinical lot available and then validate your assay using that clinical lot. The problem with this one is that you could have major delays happening if you're not validating the assay, you're putting all of the assay creation on hold, and then that truncates your timeline between when you can start the lab work on the assay and when you can run the samples. So I've seen that become a major pinch point for a lot of our clients that are at this transition. So some strategic considerations need to be included in how you want to approach that transition and validation of the assays. All right, so to summarize, I provided you with a lot of examples there of some of the considerations that can help you to make decisions on how to prepare the assay and proactively address some of the things that you need to address to make that transition from preclinical to clinical with the PK. So to summarize, the first is you need to be able to select a method that's appropriate for human studies. So as I mentioned, there are these generic assays available. They are not appropriate for the human studies. And so that's the first decision. What method are you going to use? What strategies, what reagents are available to build that assay at the preclinical stage? And is that going to be appropriate for human studies? One part of this is, of course, the range of your assay. So that's my next bullet point here, which is choose a dynamic platform. So where possible, if you're developing a preclinical assay, the bigger the range and the better the sensitivity at the preclinical stage, the more likely it is that it's going to be appropriate for human studies. So even just getting a feel for what that is, and then as you move closer and closer to that IND submission, you're going to have an idea of what that first in human dose will be. Then you'll be able to know, based on that data, if your assay is going to perform appropriately for those human samples. And then the third bullet point, which is probably the most contentious, is start your reagent generation early. So again, this is a number of points at which the reagents feed into the assay design. But the earlier that you can start that process and have those reagents available, the more time you've built in and the easier it is to move into that clinical study phase. And then the biggest part that I will emphasize here is to evaluate things early. So the more data you can gather at those early stages of development using your human matrix, the more prepared you'll be for what needs to be done to the assay to get it ready for clinical samples. And a lot of that work can be done in tandem, so it's not necessarily inflating the scope of your work that you're doing at the preclinical stage. It's just a matter of strategically including some of those human samples in the evaluations that you're already doing. Sometimes this can even be just a few extra wells on a plate. Other times it could be an additional plate. It really depends on how you build that in, but it shouldn't be a lot of extra work. And in return, you'll have that data to be able to plan efficiently for moving the assay to the clinical stage, and that will be a great reduction in work at that end. So overall, a reduction in timeline, a reduction in the amount of resources that are put towards it. So I do have a few slides to show what this would look like for an assay that we've taken those strategies that I just outlined. So this is an example of a PK assay. It was going to be used in 2 different animal species as well as humans. And so we started out very early incorporating some of that information that I've shared. So building a specific assay, making sure we were paying a lot of attention to the range, as well as to the sensitivity of the assay, and trying to be predictive and strategic about the way that we designed the animal study. PK assay in order to be sure that it would be appropriate for humans. So in this example, this is a drug that was detected by capturing it with the target and then detecting with a specific polyclonal. And you can see for the preclinical assay, across both species, it actually performed very similarly. The range was a little bit different, the sensitivity slightly different, but we were able to use the same MRD for both and the same overall methodologies for both. And so that performed really well and captured the range that we needed for the samples. It actually was a bit more sensitive than we needed, to be honest. And then moving into the clinical assay, because we had evaluated the human matrix in the assay very early, we already knew that it would perform reasonably well. There was a bit more background, so the sensitivity is a bit higher. The range is still as big of a spread, but also shifted a little bit higher. We actually used the same blocking, those same incubations, and that sensitivity was appropriate for what we were looking for with the assay. So because of the ability to test this very early on, determine some of our reagent concentrations and other conditions of the methods with that additional matrix present, we were able to significantly lower the cost and time that was involved in transitioning that assay into the clinic. All right, so moving on to immunogenicity assays. So similar layout to what I just showed you for PK assays. So first let's talk about when immunogenicity assays are implemented at the IND enabling preclinical and at the clinical phase, what do those look like compared to one another? So the first thing that's important to consider are the regulatory expectations around these types of assays. So ADA, sometimes called TAB, ADA, TAB, or NAB assays, the immunogenicity assays that are classically used for these types of programs, may be implemented at preclinical. So I stress that because sometimes if there are appropriate PD markers, if there's a sufficient body of information provided by other assays, ADA assays may not be performed at the preclinical stage. A lot of that is very therapeutic specific and study design specific, but it's pretty much expected if you take a human protein or a human antibody and you put it into an animal, you will get some type of immunogenicity response. So it just becomes a matter of what that data is needed for. When it is implemented at the preclinical stage, often that data is meant to provide context for something else, specifically the PK, for example. So if you see a change in the PK profile, is that driven by an ADA response? Because of the use of the data at that stage, this is something that is typically not a regulated assay. Often people include them just as secondary or even exploratory endpoints, not really a key aspect of what they're evaluating at the preclinical stage. Whereas when you reach the clinical stage, it becomes a safety and efficacy endpoint. It's much more scrutinized, so the data is more important within the context of the clinical studies, and so you'll have regulated ADA or validated ADA assays. Now, that being said, there are still some assays that are validated at the preclinical stage, and I'll get into some of the details of how that compares between the two stages in just a moment. The other thing that came up with PK that of course is still going to be an issue with immunogenicity assays is that when you're moving between animals or from animal into human, you're going to see changes in the matrix effects for various reasons. And then, of course, the drug and reagent lots are an issue too. So to talk a bit more about the regulatory expectations, when you are using a validated assay at the preclinical stage versus the clinical stage, there are some clear guidelines around that. And so I have some of the documents listed here that discuss those assay approaches and how an assay should be validated. But the one I'm going to focus on really is this last one, which is a publication from an EBF working group. The reason I want to focus on this is because it specifically outlines what is the appropriate strategy for non-clinical immunogenicity assessments, non-clinical ADAs. And this is something that has been implemented across the industry. So everything that's in this paper has started to become incorporated in the general handling of these types of assays for everyone in the industry. And the FDA has seen this. There's been a lot of interaction with regulatory agencies, and it's become an acceptable practice. So in that paper, in this table, that we've adapted that information into a table so you can see the parameters that are evaluated across the validation, and you can see how they differ between the non-clinical and clinical. So I'll summarize it by saying, for a non-clinical validation of an ADA assay, overall, it's a much smaller scope than what you for the clinical. It's not that parameters aren't necessarily evaluated, the key ones are still there. It's just that the overall scope of that work is smaller, it's a bit more streamlined, a bit more specialized, and that again is because of what the data is being used for and what makes the most sense when you're looking at something in an animal versus in a human. One thing I'll point out is in the top, it says the parameter tiers. So a typical tier system for an ADA is a 3-tier system where you're doing a screening assay followed by a confirmatory followed by a titer assay. So that's clearly outlined for clinical assays, but when it comes to non-clinical, there's a bit more creativity around the application of those different types of tiers. So some people do just a screening assay only and then may use the signal to noise from that to approximate a titer. So it's not necessarily following that traditional 3-tier system. The next thing that I want to talk about when it comes to regulatory expectations is around the surrogate positive controls. So these are your positive antibodies that are used to mimic a positive patient response to evaluate the performance of your ADA assay. There's very clear guidance for what the expectations are around the clinical SPC for an ADA assay, and I've listed all of this here from one of the FDA guidance documents about what's appropriate. You need a antibody that's generated against your drug. It needs to be affinity purified. It can be monoclonal or polyclonal, but it does need to be specific to your drug, and you can read through all the other details that are listed here. So that's for clinical. But when we get to the preclinical or non-clinical, if you go through some of these documents and look at the things that specifically are stated, there's really not as much, basically not any, direct guidance on exactly what's supposed to be used for ADA assays when it comes to the preclinical space. So because that's loose, some people do follow the guidance for the clinical ADA assays for the SBCs. Other people get a bit more creative. So I certainly have seen a lot more variety in the way that people approach a positive control when it comes to their preclinical or non-clinical assays. So using things like a known positive animal serum sample and then using that as just a control, a positive control in their plate. Some differences there that you can consider as well. As with PK assays, I mentioned the matrix effects can be quite different when you're getting into the assays for antibody responses as well. One of the strategies for trying to mitigate that proactively when you're at the preclinical stage looking to move into the clinical stage is to consider sample pretreatments. So I have those listed over there on the right. These can be a bead, speed, ACE, heat treatment if you wanted to destroy a target potentially that's interfering, for example, or a PANDA method. All of these are focused on either removing something that is a potential interferent in the assay or purifying the antibody itself from the samples. Either way, what you're potentially doing is removing components of the matrix from the assay system by the time you get into the wells of a plate, for example, which is what's shown here in this picture. And because you're doing that, you're actually reducing the overall impact of the matrix on the assay. So if you do this with an animal sample, and then you also do the same method with a human sample, that sample pretreatment is actually going to make it much more likely that your matrices are going to perform similarly because you are removing all of those additional components that could be different between those two matrices. And I do want to point out that no matter what, when you get into the ADA assays, moving between different matrices or even moving between different cohorts once you get into clinical studies from normal healthy volunteers into a disease state, you can have very different performance of just negative samples. So your control samples that are used to set your clinical cut point or your preclinical cut point. So a cut point is the signal in an assay that's expected to determine what is positive and what is negative. So it's a cutoff between the positive and negative values. And that's established by taking normal, in quotations, normal individuals, running them, and then doing statistical evaluations. And because of this, no matter how much you plan, and no matter how much you minimize matrix effects, no matter how focused you are on making the preclinical and clinical assays the same, you're still going to have to go back and reevaluate the cut point. So it's rather unavoidable. You do have to make sure that cut point is appropriate and that in relation to the cut point, your selected concentration of your low positive control is also appropriate. So those are very specific measures for an ADA assay that no matter what you do, you're still going to have to evaluate them and demonstrate that they are set appropriately for your assay. So I did want to point out that that is a key consideration and that's not something you're ever going to be able to avoid. But to summarize those considerations, to suggest how you may incorporate solutions to be able to streamline this process, the first suggestion that I would have is because the validation activities for an ADA assay are smaller overall in scope than what you would do in a human clinical study, it's important to treat that evaluation at the preclinical stage almost as a pre-validation for human. You don't necessarily have to redesign the entire assay just because you didn't validate it the same way that you would later. That validation should give you a really good idea of how the assay is performing, even if it is smaller in scope. I don't think you should be treating it any differently, but I do think that data is important and should help feed into some of your decisions when you're getting ready to move to the clinical stage. The other thing is to consider the differences in the tiers. So as I mentioned, that 3-tier system, the screen confirmed titer, is what you'll need when you are using an ADA assay at the clinical stage. If you're just using a screening assay at the preclinical stage, you can still take a look at the data for the signals to noise. You can still get an idea of what that spread of signal looks like across your samples. So there are other ways to use that data to still help you be more predictive about the performance of the other tiers when you get further into the clinical stage, but just something to keep in mind. And then of course the SPCs. As I mentioned, you don't necessarily have to have a specific antibody control for your preclinical studies, but it's a good idea to have it. And if you don't, you should be generating it as soon as possible. Like the reagents that are used for PK assays, these are the same types of antibodies. You need time to generate them. If you wait too long, then you're behind the ball and you're trying to catch up and you can't really rush the process of developing those antibodies. So there's still going to be that time involvement. So if you can start generating those earlier, then you can use them in your assay and get a better idea of how it's actually going to perform with that SPC once you get into the clinical stage. And then of course the differences in circulating target of the drug or other interference that can be present in the matrix can be evaluated if you use human matrix and run that very early at the preclinical stage while you're developing your animal ADA assays. And then finally, I think that the number one strategy or the number one part of your strategy that you need to have is to just do an overall risk and consideration of how you're developing the method. So do you have a sample pretreatment? If you don't, there's a higher possibility that your matrix is going to have a greater impact moving to humans. Are the reagents that you're using appropriate not just for research use, but also for that regulated assay use when you move into the clinical stage? And then of course the SPC, always it comes back to the SPC. That's something that you definitely need to consider as you're developing the methods. In conclusion, we went through a lot of detail about the PK assays, a lot of detail about the immunogenicity assays, including both ADA and NAB applications. Really, the overall goal here is to consider the PK and immunogenicity, which are critical for all of these drugs being developed, as a source of timeline and cost impact that can be focused on so that you can streamline these transitions, so you can reduce the timeline and cost, and really generate a more rapid turnaround of your timelines as you're bringing your drug to market. Thank you so much for joining me today for this discussion. If you have any other questions, my information is on this slide, and we would be happy to talk to you further if you would like to visit our website or reach out through any of the links provided on this. Thank you so much.